The overall goal of the following experiment is to derive lentiviral-based reprogrammed factor-free human-induced pluripotent stem cells and convert these cells into putative clinical-grade conditions. This is accomplished by first reprogramming adult dermal human fibroblasts with a single excisable lentiviral stem cell cassette, or STEMCA, into HIPSCs. Next, the reprogrammed HIPSCs are exposed to adeno-Cre-Puro-R virus for full excision and removal of STEMCA from the host genome. Then, genomic DNA extraction from the feeder-free HIPSCs followed by agarose gel analysis confirms proper STEMCA excision. Finally, the factor-free HIPSCs are converted into putative GMP-grade conditions by a slow conversion methodology into fully defined xeno-free conditions. Ultimately, viral-based reprogramming of human adult dermal fibroblasts and full conversion of the HIPSCs into clinical-grade conditions underscores the broad applicability of this methodology for reprogramming adult human cells and converting them into clinically relevant conditions.
The implications of this technique extend toward clinical applications of human-induced pluripotent stem cells, as they are completely factor-free and converted to clinical-grade conditions, making them compatible with human therapeutics.
Visual demonstration of this method is critical as the reprogramming, excision, and clinical-grade conversion steps are difficult to learn, because of the many intricate and specific procedures that must be followed to yield factor-free and clinical-grade human-induced pluripotent stem cells.
To begin, thaw adult dermal human fibroblasts.